molecules to analyze your protein-protein interactions. And what you do now is then that you take your FRET-labeled cells, you put them, as you can see over there, in the max quant, which is a flow cytometer produced by Miltenyi Biotec. And then you simply have to choose the FRET express mode and press on start, and then it runs automatically your whole analysis and gating and FRET calculation. So finally, the program produces results for FRET. They're really based on cell-by-cell basis. So you have a FRET result for each single cell you analyze in your high-throughput screening. And I already validated the FRET program. So I analyzed standard beads, which have standard linker lengths between the donor and acceptor in FRET. And I compared my FRET program to established non-automated calculations, which were developed by Förster in 1998. And if you see, there's quite a high correlation between the two methods, between the program and the manual calculation. And so you can conclude that you can really rely on the results of the program which come out here. And so I knew I could look for an application. And in this case, I analyzed the clustering of the CD3 and CD4 receptor on T cells. And I activated my cells with staphylococcal enterotoxin B, which is quite a strong activating toxin. And if you take a look at the FRET efficiencies, you see that there's already some FRET efficiency available for no activation, because CD3 and CD4 are really highly expressed on the cells. And as soon as you put your activating toxin on the cells, you see quite a strong increase in FRET efficiency, which is really significant after 2 minutes, because this is the time all the activation takes place. And if you activate your cells longer, for 5 hours with the toxin, then there's again a decrease in FRET efficiency, because the receptors are internalized into the cells. And for this reason, FRET disappears. And to validate this, I compared the results of the program to colocalization on the confocal microscope. And I normalized the results and compared the different efficiencies I got. So again, you see that for no activation, there's a lower colocalization of FRET efficiency, which is quite similar for the microscope and the FRET program. And after 2 minutes of activation with SAB, you see a strong increase in FRET efficiency and colocalization over here. And again, if you activate your cells for 5 hours, there's a decrease again in FRET or colocalization on the confocal microscope. And if you take a look at your cells under the confocal microscope, you can see the distribution of the CD3 and CD4 receptors on the cells. You can see CD3 in green because it's stained with an FITC conjugate, and CD4 in red because it's stained with an APC conjugate. And for no activation, you see that all your receptors are quite equally distributed over the cell surface. And after 2 minutes of activation, there are small clusters beginning to form, because those are the small microclusters which are later needed to activate your cells. And if you take a look at the cells again after 5 hours, you see that huge immunological synapses have formed, because this is where the T cell actually meets the antigen-presenting cell. And as a further application, I also analyzed CD3 homoclustering. For this case, I labeled the CD3 receptor on cells with either my donor and receptor FRET conjugate. And so as soon as the CD3 receptor starts clustering, we will see an increase in FRET efficiency. And again, we have a very low FRET efficiency if there is no activation of the cells. And after a short activation with the SAB, we see a really huge increase in FRET efficiency for the CD3 clustering again. However, if applied a blocking antibody, either to the SAB, so SAB blocking antibody before the activation of the T cells, which you can see here, or at the same time with the SAB to the cells, you see that there's really a Yeah, the FRET efficiency increase is not as huge as you can see for the SAB only. So you can see that the T cells are really activated by the SAB and that this effect can be blocked by a blocking antibody, and you see those results again in your FRET results. So, summing up, we developed the FRET Express Mode program, which is really much faster than the conventional methods like microscopy or QIP or the yeast-to-hybrid system. You can perform high throughput screenings because it's on a flow cytometer, and you can analyze thousands of cells within a second. It's relatively cheap, as long as you have your flow cytometer anyway, because you can simply use antibody conjugates. You need a very short preparation time if you use antibody conjugates, because you simply have to incubate and that's it. And as it is now automatic, it is easy to use. You do not have to do all the calculation steps, which are really complicated, but you simply press play and then the gating, and also the calculation is done by the program automatically. And this program is really universally usable for different biological systems, so not only for the T-cell activation, but you can also study pathway activations, and you can study protein-ligand interactions, which might be really interesting for the identification of new pharmaceutical drugs. So I would like to thank you for your attention, and I would especially acknowledge my PhD supervisors at Miltenyi Biotec and the University of Bonn.
Thank you very much, Kerstin. It's very nice work. Questions, please? They're a very shy bunch today. Just a quick question. You say application for high throughput screening. Have you run any compound sets with this technology?
To run compound sets, so we're still looking for protein-ligand interactions. To really test this on the flow cytometer. But until now, of course, the T-cell analysis were high throughput because they were on the flow cytometer, and this definitely should to work quite good.
So you have no idea of hit rates or false positives?
Not right now, no.
Okay.
Still working on that.